where does it happen, and what is the composition of the lipids in the diet? So major dietary lipids are in three forms. One is a triacylglycerol, second is a cholesterol, and third is a phospholipids. So triacylglycerol, that is TAG, is the major dietary lipid. Cholesterol is present as a cholesterol ester, and phospholipids are in form of structural membrane lipids. Okay, so daily lipid intake is around 20 to 30 grams per day, but in Western diet it may be 2 to 3 times higher than the whatever is there in the Indian diet. So what are the enzymes which are involved in the digestion of the lipid? So there are two enzymes in the initially. One is a lingual lipase and gastric lipase. So they are in the mouth and in the stomach. You have lingual lipases in the from the salivary gland, and gastric lipases from the chief cells of stomach. Alexa, stop. So from the salivary glands, you have the lingual lipases. The optimum pH is 2.5 to 5, and they are responsible for digestion of the short chain triglycerides. While gastric lipases, they are coming from the chief cells of the stomach. Optimum pH is 5.4, and they are responsible for the digestion of the triglycerides. Okay, so lingual lipases remain active in the acidic stomach environment. We have seen with the carbohydrate that there was a amylase, but that amylase was not active in presence of gastric acidity, while lingual lipases will remain active in the acidic stomach environment. Okay, so particularly important in newborn infants, it acts mainly on the short chain fatty acids, which is found in the milk, butter, and ghee. Gastric digestion is responsible for almost 30% of the triglycerides in the will be digested in the stomach. Okay. Now coming to the digestion of the lipids in the intestine. Start with the digestion of the lipids in the stomach. There are two main enzymes which are involved. One is a lingual lipase and second is a gastric lipase. Lingual lipase is coming from the salivary glands. The optimum pH is around 2.5 to 5, and it acts on the short chain triglycerides. And second enzyme is a gastric lipase. It is coming from the chief cells of the stomach. Optimum pH is 5.4, and it acts on the triglycerides. Okay. Now a few points to remember. One, lingual lipase will remain active in the acidic stomach environment. We have seen with the carbohydrate, there was amylase enzyme, okay, which was not active in presence of acidic stomach environment, but here lingual lipase will remain active in the acidic stomach environment. Particularly, it is important in the newborn infants. It acts mainly on short chain fatty acids, which are found in milk, butter, ghee, etc. So gastric digestion is responsible for up to 30% of the triglycerides in the stomach. Now coming to the digestion of lipids in the intestine. So most important step in the intestine is the emulsification of the lipids. Okay, this emulsification is very important in the process of digestion of the lipids. What is this emulsification? It is the process by which large fat globules are broken into small droplets, increasing the enzyme action. So conversion from large fat globules to small fat droplet is important because enzymes will act in a better way. Okay, so what exactly it does? It reduces the surface tension so that enzyme can access these lipid very easily. It also increases the surface area, so faster lipid digestion. Okay, so what are the factors promoting emulsification? Most important is the bile salts, second peristalsis, and third is the phospholipids. The bile salts will have detergent action, the peristalsis will help in mechanical mixing, and phospholipids will stabilize the fat droplet. Okay, so what is the role of bile salts here? There are two important bile salts. You remember sodium glycocholate and sodium taurocolate. They are important in the process of emulsification. Okay, so they reduce the surface tension of fat globules, convert large fat globules into small emulsified particles, and increase the surface area for better enzymatic action. Okay, now lipophilic enzymes in the intestine. So in the intestine, there are lipophilic enzymes. These are mainly the pancreatic lipase, then cholesterol esterase, and phospholipase A2. Okay, these are all three are coming from pancreas. Pancreatic lipase, cholesterol esterase, and phospholipase A2. So pancreatic lipase will hydrolyze triacylglycerol. Cholesterol esterase will hydrolyze cholesterol esters, and phospholipase A2 will hydrolyze phospholipids. Okay. Now, what is the role of the bile? The bile, the pH will be maintained around 7.7, which is more alkaline, and that neutralizes the acidic chyme. and it results in optimum pH for pancreatic enzyme action. So main digestion is happening in the intestine, and for that optimum pH is required around more towards alkaline side, and this pH is maintained mainly because of the bile. Okay, so this bile helps in neutralizing the acidic chyme and giving the optimum pH for the pancreatic enzyme action. Okay, so let us see the stepwise hydrolysis and the digestion of the triacylglycerol. So there are steps. The first step is lipase hydrolysis position 1 and 3, and they will form 2 monoacylglycerol plus fatty acids. Remember, these fatty acids are present in position 1, 2, and 3, out of which position 2, they are not able to act. So they act on position 1 and 3, and you get a 2 free fatty acids form. So you get a formation of 2 monoacylglycerol and 2 fatty acids. But still, this free fatty acid at position 2 will remain. Then comes the isomerase enzyme in action. What they do, they shift the ester bond from position 2 to position 1. Because position 2 is not accessible, so they will just shift the ester bond from position 2 to position 1. Okay, so you get a 1 MAG formed, and finally again lipase will hydrolyze this 1 MAG. You get glycerol plus fatty acid. Okay, so that is ultimately you are getting formation of the glycerol and fatty acid. But major formation is 2 monoacyl glycerol, which is around 78%. 1 monoacyl glycerol is around 6%, and free fatty acid is around 14%, and glycerol is formed in a very small amount. So these are the final products of triacylglycerol digestion. Okay, so there is a stepwise hydrolysis. Okay, the first is hydrolysis at position 1 and 3. The position 2 cannot be accessed, so there is a isomerase action, and that will shift this free fatty acid from position 2 to position 1. Then again the position 1 will be acted, and you will get 2 MAG. Okay, so and finally the product will be glycerol plus free fatty acids, and the main product is a 2 monoacyl glycerol, which is around 78%. Okay, so digestion of lipids is still partial. Okay, so then there is a digestion of the other lipids. We have seen it is in 3 forms. We have just seen triacylglycerol, what happens. Then there are cholesterol ester, which will act on cholesterol plus fatty acid, will be acted by the cholesterol esterase. Okay, then you have phospholipids. So phospholipid hydrolysis will take place in with the help of enzyme phospholipase A2, and product is a lysophospholipid plus fatty acid. So you have triacylglycerol digestion, you have cholesterol esterase, and you have phospholipids also, and there are various enzymes that we have seen. But apart from pancreatic lipase, so pancreatic lipase do not act directly, they require colipases. So there is a very important role of the colipases. So they facilitate the pancreatic lipase activity. The site of action is oil-water interface. They are secreted as an inactive enzyme in the form of zymogen. Okay, and activated by the trypsin. So there is a very important role of colipases along with the pancreatic lipases. Okay, so remember this also, that colipases are also important along with the pancreatic lipases. So importance is that it is essential for binding of lipase to triglyceride droplets. Without colipase, pancreatic lipase cannot function efficiently. Okay, so now we have seen 3 different sources, components of the fatty acid in the diet and how they are absorbed. Okay, now let us see the absorption. Okay, we have seen the digestion. Now let us see the absorption. So absorption of the long chain fatty acids. There are long chain fatty acids, there are medium chain fatty acids, there are short chain fatty acids. Okay, now fatty acids which have more than 14 carbon atoms are labeled as a long chain fatty acids. Okay, so digestion products will enter. Now we have seen there are different digestion products. Okay, there are 4 products we have just seen, that 2 MAG, 1 MAG, free fatty acid, and glycerol, a very small amount. So these are the main digestion products. Now these products need absorption. Okay, so how and what is going to happen with this? Okay, so let us see. Okay, so what is the absorption pathway? So digestion products will enter into the intestinal cells. Okay, this is by the diffusion or the micelle transport. So there is a simple diffusion or the micelle transport is there for this lipids which are there. Then what happens? There is a reesterification to triacylglycerol. So initially there was triacylglycerol which was broken down into 2 mag, 1 mag, etc. But in the enterocytes, again it will be converted into TAG by the reesterification. So this happens inside the enterocytes. Then the chylomicron is formed. It is nothing but the transport form of lipid. Okay, so lipid transport particles, that is the final package which is to be transported, that is getting formed. So that is transported via lymphatics and that will enter the blood stream later on. Okay, so what we have seen, there was a triacylglycerol in the diet. Thus triacylglycerol is broken down and various enzymes were there, and ultimately you get 2 mag, 1 mag, and then they enter into the enterocyte. But in the enterocyte again they will form the TAG, and this TAG will be packed in a form of chylomicron. That is the final packed form in which it will be transported. Okay, so that what is the transport route? The long chain fatty acids will be transported via lymphatic system, and short chain fatty acids will be transported via portal blood. Okay, so chylomicron formation, it gets formed into the intestinal mucosal cells. It's composed of triacylglycerol plus cholesterol plus phospholipids plus apoproteins. Okay, and that is the transport form of dietary lipids. Okay, so you should remember that pancreatic lipase is the most important enzyme for fat digestion. Bile salts are essential for emulsification. Co-lipase activates pancreatic lipase, and long-chain fatty acids are absorbed via lymphatics, while major product of triacylglycerol digestion is the 2-monoacylglycerol. So this is what is the we have seen up till now. The same thing is given in this table also, that lingual and acid stable lipases in the mouth. Triacylglycerol with short chain and medium chain fatty acids will form free fatty acid plus diacylglycerol. Pancreatic lipase plus co-lipase in the small intestine, they will act on the TAG with long chain fatty acids, and free fatty acids and 2 mag will be formed. And intestinal lipase along with the bile acids will act in the small intestine, and TAG with medium chain fatty acids, and they will form 3-FFA and glycerol. Okay, then phospholipase A2 plus bile acids in the small intestine will act on phospholipids with unsaturated fatty acids and fatty acids on position 2, and it will form the unsaturated free fatty acid and lysolecithin. So this all we have seen. Okay, so in short, there is a major digestion and there is a minor digestion. Minor digestion takes place into the mouth as well as stomach. So minor digestion of triacylglycerol in the mouth and stomach is by lingual and acid stable lipases. Major digestion of lipids is in the lumen of the duodenum and jejunum by pancreatic lipolytic enzymes. Bile acid facilitates formation of mixed micelle. There is a passive absorption of the products of lipolysis from the mixed micelle into the intestinal epithelial cells. Then the process of reesterification is there. 2-monoacylglycerol with free fatty acid inside the intestinal enterocyte will undergo reesterification and assembly of chylomicrons containing ApoB48, triacylglycerol, cholesterol ester, and phospholipids, and they will get exported from the intestinal cells to the lymphatics. So after after the esterification, there will be export via the lymphatics. So what happens in the pancreatic deficiency? There is a steatorrhea, and unsplit fat is present in the stools. Okay, when bile is not available, absorption is defective, split fat is present in the stools, defective absorption of the vitamin K leads to the prolonged prothrombin time. Okay, so there are 6 major steps of the lipid absorption. Minor digestion of triacylglycerol in the mouth and stomach by lingual lipase, major digestion in the mouth and jejunum by pancreatic lipolytic enzymes, bile acid facilitates formation of mixed micelle, formation of passive absorption of the lipolytic products into the intestinal epithelial cell is happening. Then there is a reesterification of 2 mag with free fatty acid, and this assembly of chylomicrons will get transported via the lymphatics. Okay, So this mixed micelle formation, what happens in the enterocyte, that is a mixed micelle are nothing but the molecular aggregates of lipid digestion products with the bile salts that facilitate the lipid absorption. Okay. So composition of the mixed micelle are 2 MAG, long chain fatty acid, cholesterol, phospholipid, lysophospholipids, and bile salts. Okay. So basically, what is the structure of this micelle? From outside, it is hydrophilic, but from inside, it has got a hydrophobic core. Okay. So it is essential for intestinal absorption of the lipids, and it is also responsible for the absorption of the fat soluble vitamin. Because it is hydrophilic from outside, it will be easy for this fat soluble vitamins absorption, and these vitamins A, D, E, and K. There are 4 vitamins. We know that these are fat soluble vitamins and they will get absorbed. Okay. So now absorption into the intestinal cells. So micelle will reach the microvilli of the jejunal mucosa. There is a digestion products diffuse into the intestinal epithelial cells, and absorbed molecule will be fatty acids, 2-monoacylglycerol, cholesterol, and phospholipids. Okay. So mechanism is by passive diffusion. Okay. Now, what happens further is the enterohepatic circulation of the bile acids. We have seen that bile acid is very important, but there is an enterohepatic circulation of the bile salts. Okay, so recycling of the bile salts between the intestine and liver is happening. The process is that bile salts participate in the lipid digestion. They are reabsorbed into the ileum. They will return to the liver via portal circulation, and they are resecreted into the bile. That is what is the uh, enterohepatic circulation. Okay. So reabsorption of the bile acid is to the extent of 95 to 98%. So re-esterification inside the intestinal mucosal cell. Okay. So once that once inside the enterocytes, free fatty acids are converted back into the TAG. This also we have seen. So there is an activation of the fatty acids. Fatty acid will get activated to fatty acyl CoA. Enzyme is a acyl CoA synthase. Okay. And this requires 2 high-energy phosphate bonds. Okay. Then finally, the triacylate glycerol is formed. So triacylglycerol to some intermediate products. From intermediate products, again you get a triacylglycerol. So you get monoacylglycerol plus fatty acyl CoA forming diacylglycerol, and this diacylglycerol plus free fatty acyl CoA will form triacylglycerol. Okay, so monoacylglycerol pathway, that is a MAG pathway, a major pathway for TAG synthesis in the enterocytes. So monoacylglycerol is the major pathway. But along with this, there is a some amount of glycerol also is getting formed. When there is a digestion, along with that, we have seen that small amount of glycerol is also getting formed. But what happens to this glycerol? Okay, the free glycerol from the intestine will directly enter into the blood. Okay, there is a whatever the glycerol which is formed from the lipid digestion, that will directly enter into the blood. But from TAG formation, in the enterocytes, some amount of glycerol is also required, but this glycerol is taken from the glucose via glycolysis. Okay, so in the enterocyte also you require some glucose from some glycerol, but that glycerol is not taken from the glycerol which is absorbed. That will directly, the one which is absorbed will directly enter into the blood, but this glycerol will be derived from the glucose via glycolysis. So you have carbohydrate utilization here, and from this glycolysis you will get the source of the glycerol. Okay. Now, the next step is the formation of chylomicrons. Chylomicrons are the lipoprotein particles that transport the dietary lipids. The clinical application is that carnitine shuttle system. You should understand that there is an important concept of carnitine shuttle system. We know that these fatty acids are short chain fatty acids, medium chain fatty acids, and the long chain fatty acids. Okay, out of these short chain fatty acids, medium chain fatty acids, and long chain fatty acids, these long chain fatty acids cannot be absorbed directly. They require some transport, and that is the carnitine shuttle. Okay, so the carnitine shuttle system is essential for transport of long chain fatty acids into the mitochondria for beta oxidation. Okay, so this is mainly for the long chain fatty acid, because ultimately it has to go to the mitochondria for beta oxidation. However, short chain and medium chain, they are directly oxidized. Okay, they will cross the membrane easily. They will have a direct access into the mitochondria, but not with the long chain fatty acids, and they will require this carnitine shuttle. Okay, so important concept is that short chain fatty acids and medium chain fatty acids can directly enter mitochondria. Long chain fatty acids will require carnitine transport system. Okay, so what is the clinical application? Medium chain and short chain fatty acids, when will not require a carnitine, there is a direct mitochondrial entry. The oxidation is rapid and efficient. So the clinical significance is that they are used in the malabsorption syndrome, and they provide quick energy source. Okay, what provides quick? Short and medium chain fatty acids, because this long chain cannot be. So when there is a malabsorption syndrome, long chain fatty acids will not be absorbed. Okay. Now another application is the carnitine deficiency. If there is a deficiency of the carnitine, which is required for the mitochondrial transport of long chain fatty acids, which are the high risk group which have the deficiency of carnitine, it is a preterm infants. Okay, so there is an impairment of the fatty oxidation of long chain fatty acids because they cannot enter the mitochondria. There is an increased glucose utilization, so energy is produced mainly from the glucose, and there is a hypoglycemia due to excessive glucose consumption. So that is also another clinical significance. Another application is a translocase deficiency. That is the defect in the carnitine acyl carnitine translocase, which transport fatty acyl group across the inner mitochondrial membrane. Okay, what is the metabolic effect? There is a defective oxidation of long chain fatty acids. Okay, then what are the clinical manifestation? In fasting state, you get muscle cramps. During exercise, you get muscle weakness, and high fat diet, there is a metabolic symptoms. Okay, so disorders of the fatty acid transport, there can be deficiency of the carnitine. So because of the reduced carnitine, there will be impaired long chain fatty acid transport. If there is a translocase deficiency, there is a defective carnitine shuttle and reduced beta oxidation, and normal or short chain fatty acids and medium chain fatty acid metabolism, carnitine is not required, so there is a normal oxidation. So take home message is that carnitine shuttle is required for long chain fatty acid oxidation. Short chain and medium chain fatty acids do not require carnitine. Carnitine deficiency leads to hypoglycemia due to increased glucose utilization. Translocase deficiency lead to muscle cramps during fasting or exercise, and preterm infants commonly have carnitine deficiency. Okay, so that finishes one of the major topic from the lipid metabolism. Now the next topic uh, is a ketosis. So we begin with very important topic for the examination. Always there'll be one or two questions on this topic, that is a ketosis. Okay, so definition of ketosis. Ketosis is a metabolic condition characterized by the increased ketone bodies in the blood, that is ketonemia, excretion of ketone bodies in the urine called as ketonuria, and presence of acetone smell in the breath. Okay, when the rate of ketone body synthesis exceeds their utilization by the extrahepatic tissue, ketone bodies will accumulate inside the body. Okay, so normally what is the level of the ketone bodies? So it is less than 1 milligram per deciliter, and urinary ketones generally they are trace not detectable by a urine test. So normally ketone bodies produced by the liver are rapidly utilized by the peripheral tissue. So where these ketone bodies are formed in the liver, and they are rapidly utilized by the peripheral tissues. So what are the types of ketone bodies? Again, this is a SBA which is frequently asked. One is acetoacetate, second is beta hydroxybutyrate, and third is acetone. Acetoacetate is the primary ketone body. Then secondary ketone body is the beta hydroxybutyrate, and acetone is the volatile ketone body. So acetoacetate is the first ketone body which is formed. Beta hydroxybutyrate is formed by reduction of the acetoacetate, and acetone is produced by spontaneous decarboxylation of the acetoacetate. Okay. Now, what are the causes of ketosis? One is the insulin deficiency that leads to increased lipolysis. Okay. There is an increased fatty acids that leads to the increased beta oxidation, and there is an increased acetyl CoA that will lead to excess ketone formation. So what happens in the diabetes? There is an insulin deficiency. Insulin deficiency is there, so glucose metabolism is hampered. There is a diversion to the lipolysis. So increase lipolysis. So fatty acids are increased. They will undergo beta oxidation. So once they undergo beta oxidation, acetyl-CoA will be produced, and this acetyl-CoA will be diverted for the formation of the excess ketone bodies. Okay. Now there are other factors that the oxaloacetate is diverted to the gluconeogenesis. TCA cycle activity is reduced, and excess acetyl-CoA will get converted into ketone body. Okay. Now what happens in the starvation? In starvation, glucose intake is reduced, so there is an increased lipolysis. There is an increased fatty acid oxidation, so excess of acetyl-CoA is formed. The oxaloacetate will be converted to gluconeogenesis, and the TCA cycle activity is reduced, so excess acetyl-CoA will form ketone bodies leading to ketogenesis. Okay, so this is an important concept. So during prolonged fasting, brain derives 75% of its energy from the ketone bodies. So apart from this, there are other causes. What we have seen, diabetes mellitus and starvation. But other causes are there. That is a prolonged vomiting. What happens in hyperemesis gravidarum? That is a starvation state. Uncontrolled diabetes leading to increased lipolysis, low carbohydrate diet, increased fatty acid oxidation, etc. Now, there is a hormonal regulation of ketogenesis. It is mainly the glucagon versus insulin. Okay, we know well that glucagon stimulates ketogenesis and insulin inhibits ketogenesis. Insulin will promote the glycogen metabolism. Automatically, the TCA cycle activity will be increased, and exactly opposite will happen when insulin is absent, and the glucagon activity will be more. Okay, so glucagon will stimulate the ketogenesis. So high glucagon to insulin ratio will promote lipolysis, fatty acid oxidation, and ketone body formation. So regulation of ketogenesis is regulated by three metabolic levels. One is the source of ketone bodies, that is free fatty acid. Second, insulin inhibits lipolysis, and third is a glucagon, which stimulates the lipolysis. So more free fatty acids, more ketone bodies are formed. So if there is a lipolysis, you will get more free fatty acids, and that will the source is more, substrate is more, so you will get more ketone body formation. Second, entry of fatty acids into the mitochondria. Now you have got these free fatty acids, but there is an entry into the mitochondria. The enzyme which is responsible is a carnitine acyl transferase 1, CAT1. Okay, entry of fatty acids into the mitochondria is under this enzyme, carnitine acyl transferase 1, CAT1 enzyme, and this CAT1 enzyme is regulated via malonyl CoA. Okay, this malonyl CoA will inhibit CAT1. Okay, so in diabetes, what happens? Glucagon level is increased and malonyl CoA activity is reduced. So CAT1 is not inhibited. Okay, so beta oxidation is increased. Okay, and level 3 is oxidation of the acetyl CoA. So normally acetyl CoA undergoes TCA cycle, but here this TCA cycle activity is low. So in digestion, in diabetes and starvation, the oxaloacetate is diverted to gluconeogenesis. TCA cycle is slowed down, and excess acetyl-CoA is getting converted into ketone bodies. Okay. So what is the pathophysiology of the ketosis? Increased lipolysis, increased free fatty acids, increased beta oxidation leading to increased acetyl-CoA, reduced TCA cycle activity, acetyl-CoA will get accumulated leading to ketogenesis and ketone body production. Okay. So clinically, what you get? Metabolic acidosis. Acetoacetate plus beta hydroxybutyrate are acidic in nature. So reduced plasma bicarbonate will be there. Bicarbonate is consumed in buffering the excess acid. Okay, so there will be reduction in the plasma bicarb. There will be small breathing, deep and rapid breathing due to metabolic acidosis. Then there will be fruity breath odor, that is due to acetone exhalation. Osmotic diuresis, ketone bodies in the urine cause increased water loss. Then there is electrolyte loss. Ketone bodies are excreted as a sodium salt. So sodium will be lost, and it will result into the dehydration because of osmotic diuresis and sodium loss. Ultimately, there will be coma. Severe ketosis will lead to hypokalemia, dehydration, metabolic acidosis. So how do you diagnose ketosis? It is by urine detection of these ketone bodies by Rothra's test. Okay, what is the principle of this Rothra's test? It is a nitroprusside reaction. So saturate the urine with ammonium sulfate, add sodium nitroprusside, and you add ammonia, you get a purple ring. that we say ketone bodies are present. So remember, from SBA point of view, Rothra's test is for the detection of the ketone bodies, and the principle is nitroprusside reaction. When you saturate the urine with ammonium sulfate and you add sodium nitroprusside, you add ammonia, you will get ketone bodies, okay, purple ring, and that will detect the ketone bodies. Okay, what is the differential diagnosis? One is a diabetic ketoacidosis, where the Benedict's test will be positive as well as Rothra's test will be positive. And what happens in starvation ketosis? Benedict's test will be negative, but the Rothra's test will be positive. That is how you differentiate from the diabetic ketoacidosis from the starvation ketoacidosis. Now coming to the management of ketoacidosis, we know that insulin will reduce lipolysis. Glucose infusion will prevent hypoglycemia. Bicarb you need to is required for correction of the acidosis, and fluid replacement will treat the dehydration, and electrolyte monitoring will prevent the complication. So this is what is the pathophysiology which is happening and how you need to correct by giving insulin, glucose infusion, bicarb, fluid replacement, and electrolyte monitoring. Okay, so important clinical point is that when insulin and glucose are given, potassium moves into the cell, so electrolytes must be monitored carefully. Okay, so take home message that ketosis is equal to ketonemia, ketonuria plus acetone. Acetoacetate is the primary ketone body. Most common cause is the uncontrolled diabetes mellitus. Starvation also causes ketosis, and high glucagon to insulin ratio stimulates ketogenesis. Oxaloacetate diversion to gluconeogenesis promotes ketosis. Plasmal respiration is typical of ketoacidosis, and Rothra's test detects ketone bodies in the urine. Okay, so that finishes the metabolism of lipids.